Alright, hello. I'm Cecilia Humber, and the title of my project for this is Degradation of Insoluble Protein Aggregates in Yeast and Human Systems. So, for those of you that are not science people, you probably have no idea what I'm talking about. So I'm going to give a little bit of background of just the basics of proteins, which will help you understand the significance and the problem of my project a lot better. So proteins are one of the most vital biomolecules for life. Your body is absolutely full of them, and they're responsible for tons of cellular functions, from structure to transport to energy production, cellular reproduction. They're super important. Proteins are composed of amino acids. That's their building block. And the order and the structure of those amino acids determines the structure of the protein. And the structure of the protein is absolutely vital for its function. It can't function unless it has the proper structure. So how do you get that structure? The best way to visualize this process, you can imagine a long string of amino acids, and these amino acids all have different properties, and the long string of amino acids that is the protein is going to condense down into the shape that you want it to be. And this happens via a process called protein folding. And you can kind of think of protein folding like folding origami or a paper airplane or like one of those cootie catchers that you play with in elementary school, where every step has to be done in the right order and very precisely in order for the protein to fold correctly and for you to have the right product at the end. And that product is called the native state protein. It's in the structure and the form that it needs to be in order to function. Now, sometimes proteins need a little bit of help to fold efficiently and to fold properly, and that's where these proteins called molecular chaperones come in, and these are just proteins that exist in cells to help our proteins fold into that native state. Now, as I said, protein structure is vital to function. Well, that structure can be disrupted, and that can happen via a process called misfolding that causes a non-native conformation. Now, the native conformation is the conformation we want that has its function, but then we can have a non-native conformation where it doesn't have the right structure and it can't actually function properly. So this misfolding is a normal effect of cellular aging. As cells get older, their proteins are just not as good at folding, or they'll fold correctly and then mess up a little bit. But this misfolding can also arise due to mutations or stressors on the cell, like elevated temperature. Now, cells don't really want a lot of non-native protein hanging out. You don't really want a lot of protein in there that's not going to do its function. What's the point of having a bunch of proteins that aren't actually going to do what you want them to do? So the cell does have pathways to degrade these non-native proteins, and chaperones can also refold them and fix that non-native structure, bring it back to the native structure. But sometimes those pathways, those mechanisms can get a little overwhelmed, and what you end up with is a lot of non-native protein in your cell, and that leads to something called aggregation. Now, aggregates are insoluble clumps of non-native state protein. Basically what happens is you have these non-native state proteins that aren't very stable because they're not folded correctly. They're not in the structure and the orientation that they like to be in. They have stretches that are exposed to the watery insides of the cell that don't like being in water. That's called hydrophobic. And so what happens is these non-native proteins, specifically those hydrophobic parts, flock together so that those hydrophobic parts that were exposed are now no longer exposed because they're in the middle of this giant clump of protein. And that's not great. These aggregates can be ordered and stable, in which case they're called amyloids, which you may have heard of, or they can be more random and unstable, but regardless, they confer a great deal of toxicity on the cell, and aggregates are even implicated in a whole family of diseases called protein conformational disorders. Now, you may have heard of some of these. They include Alzheimer's and Parkinson's disease, so clearly aggregation is not good. Well, you may be wondering, why don't the molecular chaperones just fix that problem? Well, this big insoluble clump is just too much for normal molecular chaperones to handle. They can't pull apart those proteins and refold them or degrade them very easily. Now, some organisms have a type of molecular chaperone called a disaggregase that can do that. And this is what my project is looking at. More specifically, there is a disaggregase in yeast that's called HSP104, and this protein we already know is able to pull apart aggregates because it's an disaggregase, but human cells don't have HSP104. So my project specifically is looking at what do human cells use instead of HSP104? What proteins might we have that function in the same way? And that's really significant because the more we know about those types of pathways, the better we can understand diseases like Alzheimer's and Parkinson's and be able to diagnose them better, understand them better, maybe eventually treat them way far down the line better. So this is very important. Now, I was supposed to be in the lab this summer doing experiments, and clearly that was not able to happen. So what I decided to do for this presentation was talk a little bit about the aims and the methods that I would have been investigating had I been in the lab, and then talk about what I actually did this summer. So my project has two aims, and I'm calling them, for the purposes of this presentation, the control study and the proximity study, in order to investigate that question of what do human cells use instead of that yeast protein, HSP104. So the control study, our lab has already made a protein, it's called GD star, that has a temperature-sensitive mutation. So above a certain temperature, it is mutated and it is prone to aggregation. So this is really good for us to study because we can control when it aggregates, and then we can study that. But we need a control for our experiments to study GD star. So we have already in the lab reversed that mutation to make a protein that we've called GD. And now the first arm of my project is to make sure that that can function as an effective control for GD star. Then the second aim of my project is to determine proteins that interact with that misfolded substrate, with GD star when it's aggregating. And I'm calling that the proximity study. It's what might be around that aggregate when it's aggregating. So I'm going to go very briefly into the methods. I know that scientific methods can be a bit complex and a bit boring if you're not a science person, so I'm going to try and break it down as much as possible. But if you have questions, feel free to leave them in the thread underneath this. I'm happy to answer them. I'm happy to answer questions in the Q&A session. So for the control study, we're studying solubility. Solubility is used to study aggregation because aggregates are called insoluble aggregates. So we can look for proteins that aren't soluble and assume that those are aggregating in this case. So what we've already done with our mutated protein GD star is we've looked at its solubility below that cutoff temperature and above that cutoff temperature when it's aggregating, and we've seen that at that higher temperature it is not soluble in detergents. We use detergents because detergents are really good at dissolving insoluble things. That's why you use dish detergent to wash your dishes, because the grease on your plates isn't soluble in water, so you use detergent to help it dissolve in the water. So we use different strengths of detergents to study the solubility of GD star, and like I said, we've seen that it's not soluble at elevated temperatures, so we know it's aggregating. So what we've already begun to do, what we were doing before the pandemic hit and what was going to be the beginning of my project, was to do those same studies with GD to see, is it soluble at lower and elevated temperatures across all of these detergents. And what we've already begun to see is that it's soluble to the same degree in the lower temperature and the elevated temperature, indicating that it's not aggregating the same way that GD star was, where it was insoluble at the higher temperature. So we still need more trials of that to increase our N before we can move on to actually using GD as a control. But once we're able to do that, we're doing a proximity study, where basically what we've done is we've taken our protein GD star and we've added a little tag on the end. And what that tag does is it causes this other protein called biotin to bind to anything within 10 nanometers of GD star. And so that means that anything around GD star, when it's aggregating or not, is going to have biotin on it. And this is really helpful because biotin binds to another protein called streptavidin. So we can use that protein, streptavidin, to separate out anything that has biotin and anything that doesn't. And then we can go on to study those proteins that have biotin on them to see what tends to hang out around GD star when it's aggregating, and study those proteins further to see if they might be disaggregases. So, since I couldn't do all that, what did I actually do this summer? Well, I am going to be writing a senior thesis as part of the departmental honors program in the bio department. So I've kind of gotten a head start on that. I've been searching up and reading journal articles and reviews about all of the topics that my project centers on protein folding and misfolding and aggregation and disaggregases and disease, all that jazz. And then I've started writing the introduction section of my paper. But I figured that it would be kind of boring for me to spend 8 to 10 minutes talking about reading journal articles and writing a 10-page introduction. So I thought that I would give you more of a background on my project and more of what we had been planning to do. Especially because my future directions, once I'm allowed to go back into the lab, hopefully around August or September, is to continue investigating those aims, and then once we have found those proteins in aim 2 that hang out around our mutated protein, then we can go on to characterize them and see if they might be disaggregases. So that's my project. Thanks for listening. Feel free to ask any questions below. I'm happy to answer them. I know that scientific research, especially in an area that you aren't confident or comfortable in, can be a lot, so I tried to break it down as much as possible. But yeah, thanks for listening.